Hello and welcome. In this video, I will be guiding you through the process of tissue culturing bananas. Tissue culture is a technique used to produce new plants from small tissue samples. It is an essential technique for plant propagation and is widely used in the production of bananas. By the end of this video, you will be able to understand the basics of tissue culture and how to apply this technique to produce healthy banana plants. Step 1. Preparation. To begin, you will need a few materials and equipment. You will need a sterile work area, a laminar flow hood, a scalpel, tissue culture media, petri dishes, and banana plantlets. Ensure that all the materials are sterile before you begin. The work area and laminar flow hood should be cleaned with ethanol to prevent contamination. Step 2. Disinfection. The first step is to disinfect the banana plantlets. This is important to prevent any contamination from bacteria or fungi. To do this, you will need to cut off the roots and leaves from the plantlets. Then, using a scalpel, cut the shoot into small pieces, about 1 centimeter in size. These pieces will then be placed in a beaker containing a sterilizing solution, such as a solution of sodium hypochlorite or hydrogen peroxide. The plantlets should be left in the solution for about 10 to 15 minutes. Step 3. Culturing. After disinfecting the plantlets, they are ready for culturing. Place the pieces of plantlet in a petri dish containing tissue culture media. This media will provide the necessary nutrients for the plantlet to grow. The petri dish is then sealed to prevent any contamination and placed in a warm, dark environment for a few weeks. Step 4. Transfer. After a few weeks, the plantlets will have grown and will be ready for transfer to a new culture medium. Using a sterile scalpel, carefully transfer the plantlets to a new petri dish containing fresh tissue culture media. The process is repeated, and the plantlets continue to grow and multiply. Step 5. Transplanting. Once the plantlets have grown to a suitable size, they are ready for transplanting. Carefully remove the plantlets from the petri dish and plant them in a potting mix. The potting mix should be sterilized to prevent contamination. In conclusion, tissue culturing bananas is an essential technique for plant propagation. It allows the production of healthy banana plants from small tissue samples. The process involves disinfecting the plantlets, culturing them in tissue culture media, transferring them to new media, and finally transplanting them into a potting mix. With practice, you can use this technique to produce healthy banana plants in large quantities. Thank you for watching, and I hope you found this video helpful.